Everyone, welcome to our third webinar on decoding genome in monogastric, the series. And today we will discuss about epigenetics. And this webinar is in partnership with GeneSwitch and the European Master for Animal Breeding and Genetics. And in that direction, I pass the floor to Gabor, who would introduce the program and what they've been doing in this joint master. Thank you.
Yeah, thank you very much, Gina. So welcome, everyone. My name is Gabor Mesars, and I work at the University of Natural Resources and the Life Sciences, Vienna, Austria. And well, we are the coordinators of the European Master in Animal Breeding and Genetics Master Program, so joint master program, the EMABG being the well, abbreviation of this program. So, I mean, I would just tell a few words briefly about this program, as we are co-organizers of this meeting. But if you are interested in, well, the same things that I told now, or even more, and in more detail, so then the bottom of this slide, you see the emabg.eu website, which has all the information that you might be interested in. But right now, just briefly, so this is a master program that is supported and co-funded by the European Union Erasmus Plus program, as it's also seen in the very prominently placed logo on this slide. And basically, it is a master program with a joint cooperation between 6 universities, as you will see on the next slide, 3 associated academic partners and 13 industry partners who are also associated partners. And the goal is to respond to the current and future needs of animal breeding community, including the industry. Hence, also our collaboration with the EFAB and other entities in this respect. So the mentioned logos of the mentioned universities of the partners. So we are from the University of Natural Resources Life Sciences Vienna. That is the abbreviation is BOKU. From the German name, but also then counterclockwise Wageningen University, University of Göttingen, NMBU from Norway, SLU Sweden, and AgroParisTech Paris-Saclay from France. And well, there are also our associated partners mentioned for which are also particularly important, especially for the students, because well, these are the primary partners where they could do internships at these places, but also, of course, other places as well. This is just a brief structure of the program. So this is a standard 2-year, 4-semester master program. First year being at one of the 2, which we call a first-year universities. This is Wageningen University or University of Göttingen, where we have a particular focus. And from here, the students, after the summer course, which is held well between the two academic years, they could go to one of the 4 study tracks. So this is for our case, at our university, we have the breeding programs in low-income countries. If they want to follow the biological and societal context of breeding, that would take place at SLU Sweden. At NMBU Norway, there is the genomic selection and precise phenotypes, and the AgroParisTech Paris-Saclay has the functional genomics study track. And well, this is well, of course, all of this has a particular curriculum attached, and with the very specific courses, and obviously a very tailor-made master thesis possibilities, also including our possibly including our associated partners, and well, many other things that you might be interested in. But well, I don't want to keep take too much time here, so I would just once again highlight the emabg.eu website, where you can find this information and more if you're interested in our master program. So this is well, there are scholarships now they run out from the EU side, but also there is a possibility to obviously attend of this master program. There is each year there is a call for applications. So whoever would like to attend, then it is the best way is to respond to these calls for applications, and then well, you can follow up from that point on and follow up the process, and also then hopefully come to us or one of our partner universities to study. So thank you very much.
Thank you, Gabor. So yeah, I would like to introduce myself again. I'm Gina Kardik, and I represent European Forum of Farm Animal Breeders. I am actually an ex-student of Gabor, so I was in the EMABG program. And if you didn't hear about it and you're interested in animal breeding and genetics, please do have a look. It's a pretty interesting program. And to come back to the EFAB, we are actually very representative of breeders in Europe. So our role is to ensure the representation of our breeders at the EU level, so both for policy and legislation level. But we are also promoting, supporting and promoting sustainable balanced breeding. So we do that through the code EFABAR, and this is represented through all our members, which is poultry members, ruminants, pigs, aquatic, and now newly the insect. So the aim of EFAB is to engage and promote sustainable animal breeding, and also provide knowledge through different EU projects, so including Gene Switch. And you can see the different projects related to the species, but also a pure project like Eurofan and Roadmap, which involves every other species. And also on the right-hand side, you can see all our members. So it's both breeding and reproduction organization and association. And alongside that, we are also, our EFAB members are also involved in our technology platform, which includes universities and knowledge centers. And the aim of FabreTP, which is Farm Animal Breeding and Reproduction Technology Platform, is to develop and research and innovation, and also to set priorities. And we connect knowledge between private and public sector. So that's why on the left-hand side, you can see all the university and knowledge center who are members of FabreTP. And yeah, so in a nutshell, we want to promote and support research and innovation in both breeding and reproduction technologies within the animal sector. And yeah, if you want to have more idea of what we do, you can go on our website. So it's efab.info, and you would go directly on our website if you go there. And yes, I would like to move on to our house rules. So please, everyone, it's important for us to mute your microphone. I would like to tell you again that this meeting is being recorded and it will be shared later on. So if you don't want your face to be shown during the record on the YouTube channel, you can switch off your camera. If you want to ask questions, you can raise your hand using the emoticons on the bottom. And if you feel more comfortable in asking questions through the chat box, this is also possible. You can just write your question and we will try to ask all of our guests today the questions. And if there is any problem and you can't access the chat box, please send an email to Maria. She will do her best to help you. So now we can start with the agenda. So the first part, we will explain the role of epigenetics in monogastric breeding, so both poultry and pigs. And then Linda will help us, will introduce influence of maternal diet on SCFA levels and microbiota. And then we will have Smahane, who will talk about the influence of maternal diet on pig epigenome and transcriptome from mother to fetus to piglet. And yeah, I will leave the floor now to Chala, who will introduce our guest. Thank you.
Thanks, Gina. Well, in fact, I'm just going to give the floor to Linda so she can also introduce herself and yeah, explain her research to us. Linda?
Yes, thank you. you are looking now at the presenter mode, right?
Yes.
Good. Okay, thank you very much for inviting me for giving a talk here today. So my name is Linda Loonen. I work at Wageningen University at the Host Microbe Interactomics group as a researcher, and I'm part of work package 5 of the Gene Switch project. And today I will be telling you something about the influence of the maternal diet on short-chain fatty acid levels and the microbiota. Smahane, who will give a presentation after me, is also part of this project. So I will give some introduction and background to this work package and share some first initial results. And then Smahane will go on to the other results that we've obtained in this project. So as you may be aware, there is a link between the diet, microbiota, and health. As you can see, let me see if I can make a pointer. Yeah. So if you have a normal functional microbiome, everything should be balanced, and there should be a healthy situation. But if you have a less diverse dysfunctional microbiome, you will have lower abundance of strict anaerobes, leading to a lower abundance of short-chain fatty acids, such as butyrate. And if you have a dysbiosis in the microbiota, you are more susceptible to disease and the severity of diseases. And some diseases you can think of are allergy, obesity, autism, type 1 diabetes, type 2 diabetes, cancer. So it's very important to have a balanced microbiota and also a balanced diet. A lot of interest has been put on these interactions between the diet, microbiota, and health lately in literature. And there are many different studies that look at different metabolites and mechanisms that connect diet, microbiota, and the immune responses. Actually, what we're focusing on are the short-chain fatty acids. So these are metabolic byproducts of microbial fermentation of complex polysaccharides, and that are not digested or only partly digested in the small intestine. These non-digestible polysaccharides are found in plant cell walls and can be further classified into soluble and non-soluble dietary fibers. And then the soluble non-digestible proteins are highly fermentable and typically lead to short-chain fatty acids in the colon. And as you can see here, there are fewer than 6 carbons in the tail. Most abundant in the intestine are acetate, propionate, and butyrate. And also those are the 3 that I will focus on in this presentation. So why do we focus on short-chain fatty acids? Well, a lot of studies have been done lately focusing on short-chain fatty acids, and these are known to be epigenetic and metabolic regulators. And as you can see here, this is just a short summary of some key papers that have been produced lately. So they see that there is some epigenetic programming in host tissues based on diet microbiota interactions, basically short-chain fatty acids. Here they can actually, they've done a study where they see an effect on type 1 diabetes. And also asthma has been shown to be dependent on the maternal diet and bacterial metabolites, which is in this case also short-chain fatty acids. So more and more interest has been shown in these short-chain fatty acids and actually their effect on the epigenome and the metabolic regulation. And that's also why we focus on in our work package. So how does that go? How does it work? Well, the short-chain fatty acids in the colon, the concentrations in the colon can be manipulated by the diet. So what you see here is sort of like a graphic representation of the small intestine and leading to the colon. So in the small intestine, there is some digestion by the host. And then in the colon, the undigestible fibers are actually fermented by the microbes, leading to the production of short-chain fatty acids, which can actually be taken up by the host and end up in the circulation. As you can see here, the human genome contains approximately 17 genes to degrade fiber. However, the microbiome encodes many genes to degrade fiber. And there are many different bacteria that actually contribute to this fermentation. So, for example, from the phyla Firmicutes and actinobacteria, these are considered to be important in initiating the degradation of the non-digestible polysaccharides. And then after that, the breakdown is continued by mainly species of the phylum Bacteroides. Acetate production is common to many bacterial groups. Propionate is actually only produced by a few dominant genera like Akkermansia, Bacteroides vulgatus and Bacteroides theta-automicron. So there are many different bacterial species and phyla that can actually produce different short-chain fatty acids from the fibers. So what is the role of the short-chain fatty acids in intestinal homeostasis? Well, there are actually many. So as you can see here, short-chain fatty acids are actually, so there is a gradient of short-chain fatty acids. The further you go down the crypts of the intestine, the lower the concentration gets. They are taken up, as I will show in my next slide, by the intestinal lining, and they end up in the portal vein where they are transported to the periphery. So there is a big effect of short-chain fatty acids on the intestinal homeostasis. So there is an increase in regulatory T cells to control the inflammation. They can induce digestive hormone production. For example, butyrate serves as a fuel for enterocytes. So these are the enterocytes. Those are the most common cell line in the epithelial lining. And they are actually fueled by butyrate. So they take it up as an energy source. But also the expression of gut defense molecules like the AMPs, for example, here that are produced by the Paneth cells, they are induced by short-chain fatty acids. So they actually, they have a lot of different functions and they are very important in the intestinal homeostasis. So what happens to the short-chain fatty acids in the colon? Well, as you can see here, these are the fibers that actually end up in the colon where they are fermented by the microbiota. Then there are many different pathways actually leading to the different short-chain fatty acids. For example, here is a couple of pathways that lead to the production of propionate. Same here, lactate and acrylate lead to propionate. So there's many different pathways leading to the production of those three main short-chain fatty acids. And then if we look, for example, acetate is transported through this MCT1 and SMCT1. Also, it can enter the cell via diffusion. Then there are also transporters that can actually transport the short-chain fatty acids out of the cell into the lamina propria. And so this is an epithelial cell, as you can see here. So this is where the short-chain fatty acids actually can enter, and then they can leave at the bottom through the lamina propria into the portal circulation, which can thus happen via any transporters. So that's active transportation, but there is also recognition by these GPCR, so GPCR43, GPCR41, and those are actually the receptors that are expressed on the intestinal epithelial cells and on specific immune cells. And they can actually then signal to different type of immune cells in the lamina propria. So as you can see here, there's different levels. This actually comes from a study. So these are the effects of short-chain fatty acids, or the concentration of the short-chain fatty acids in a study that they did where they actually had control animals. They treated them with antibiotics, and then here they treated them with antibiotics and short-chain fatty acids. And here you can see that there is some of the levels of the short-chain fatty acids in serum, ileum, cecum, and colon. As you can see, the levels vary a lot, and in the intestine, the levels are much higher than in serum, which actually makes sense because it's processed by the liver. These are levels of concentrations in pigs, and actually in humans and in mice, for example, the concentrations are very different. So in humans, it's much lower. So that depends on species, but also, of course, on type of diet and type of study and when the timing of measurement. So then also some recent publications came out where they actually saw an effect of short-chain fatty acids to improve the meat quality and lipid metabolism in grown pigs. And also that the short-chain fatty acids could prevent fat deposition in pigs. So that actually interested us also in the Gene Switch project, that there could be an effect on the meat. And also it has been shown that there is could be an effect of short-chain fatty acids on organs and tissues beyond the gut, including the developing fetus. So this picture from a very nice review by our group actually shows what can all happen. So the maternal diet is rich in fibers. Then the short-chain fatty acids are actually produced in the gut. So they're taken up and they are transported into the portal vein. And by that, they go further into the periphery, where they can actually have an effect on the Tregs and immune regulation via FOXP3 in the lungs. They can have signaling in the pancreatic beta cells, having an effect on type 1 or type 2 diabetes. There can be some epigenetic imprinting in bone marrow, hematopoietic stem cells, and as multiple papers have shown, that there can also be epigenetic imprinting in tissues in utero. And that's an important component of the complex interactions between the diet and the animal phenotypes. So mainly, I'm sorry. Yeah, so there are many studies that actually show that there is an effect on the offspring, and that's actually what we're going to look at later on in our work package. So short-chain fatty acids can help alter the host epigenome, and the epigenome describes the modifications of to the genome that do not affect the DNA sequence, but lead to altered gene expression. Epigenetic modifications include DNA methylation and histone modification, which alter how the DNA is packed into the chromatin. So what has been described often for butyrate, for example, is that it's an inhibitor of histone deacetylation activity based on a report that describes the accumulation of acetylated histones in butyrate-treated cancer cells. And the regions of histones that undergo post-translational modifications, such as acetylation or methylation, which can be depicted in the pictures here. And the other panel depicts the consequence of condensed or more open chromatin accession, chromatin structures for gene transcription. So when the chromatin is very condensed, there is no room for DNA or gene transcription. And when the chromatin is has a more open structure, then actually transcription is switched on, basically. So this is a nice summary, basically, of the histone acetylation and epigenetics. So there is a chromosome with the chromatin. Histone is present in the histone tail. So if there is acetylation, the DNA becomes accessible, the gene will be active. If the DNA is inaccessible, the gene will be inactive. And the question was basically now, can we alter this project by having an increased short-chain fatty acid concentration in the mothers? So that was the aim of this work package, is to investigate the possible role of the short-chain fatty acid as an epigenetic regulator of liver and muscle metabolism and function impacting on skeletal muscle and body fat. The reason we chose the liver and the muscle in this particular work package, liver was chosen because it metabolizes acetate, propionate, and butyrate from the portal circulation. So once it's taken up by the gut and put into the portal vein, it initially passes the liver before it goes into the periphery. So the liver is the first organ that actually comes across these short-chain fatty acids. And we looked at skeletal muscle also because it was previously shown in literature that in sheep fetuses, the muscle can be epigenetically modulated by diet. So that's why we wanted to see what we could discover from increased diets or increased short-chain fatty acid in the mother. The hypotheses that we had in this work package was that the high fiber diet will increase serum concentration of acetate associated with the changes in microbiota. The maternal diet will have effects on the fetus in utero via short-chain fatty acid exchange in the placenta and epigenetics mechanisms. The effect of genetic imprinting in the maternal diet will have phenotypic effects in the offspring up to at least 8 weeks postpartum. Serum acetate will regulate PPAR alpha and the lipid metabolism, reducing hepatic lipids. And serum acetate will upregulate fatty acid oxidation pathways in liver and muscle and protein metabolism in muscle. So the anticipated impact of this work package is to increase knowledge about the effects of non-digestible fiber on fecal and serum concentrations of short-chain fatty acids, to increase knowledge about epigenetic effects of short-chain fatty acids in utero and potential phenotypic effects postpartum, to gain knowledge on the impact of high fiber diet and circulating short-chain fatty acids concentrations on the liver and skeletal muscle metabolism and their function. We wanted to make a biorepository of tissues for follow-up studies. And we wanted to gain knowledge on to support the use of dietary fiber to promote the pig health. So what was actually the setup of our study? So we've used 3 different groups of sows that were all on a different diet. So we had a high fiber diet, we had a high fiber plus acetate diet, and we had a low fiber control diet. In total, we had 21 sows, so 7 sows per dietary group. And they went through 2 rounds of pregnancy. So the first pregnancy actually led to delivery of piglets. Of those piglets, of those litters that were born, we took 2 females and 2 males of each sow, and we followed them up to 2 months post-weaning. Then they were sacrificed, and we collected the samples that are depicted here. So we took small intestine, also colon, heart, distal lung, kidney, and bone marrow cells, and also liver and muscle, as mentioned before. And those liver and muscles were actually used for RNA-seq and ATAC-seq. And the other samples were stored and are still stored for a biorepository. So after the first pregnancy, the sows were inseminated again. And then we harvested the fetuses day 70 post-fertilization. So the sows were euthanized in the 2nd trimester of their pregnancy. And then we took again 2 males and 2 females fetuses of each sow, where we took exactly the same samples to compare the in utero and post-weaning conditions. So we took also feces and blood from the sows on different time points, and we took samples of the offspring to measure the short-chain fatty acids and fecal microbiota. And yeah, I will now go into some more details. So just as an overview, the diets that we used. So this is the low fiber diet, a high fiber beet diet, and a high fiber pea diet. So these are 2 different plant sources that lead to different concentration of short-chain fatty acids. So in this diet, we had a lot of beet pulp added to the diet, and in the high fiber pea diet, we had a lot of pea fiber, and we also added some extra sodium acetate as sort of a positive control to make sure that there was plenty of sodium acetate present. And here you see a timetable of the experiment. So the sows were put on our intervention diets just before insemination on day 0. On day 0, we also sampled blood and feces of the sows. So on day 30 of the first pregnancy, we had blood and feces sampling. On day 70 of the first pregnancy, we had blood and feces sample. So this was the first gestation, which led to a full-term pregnancy. After delivery, the sows, or just before delivery, the sows were actually put on lactation diet because the diet we gave them would not produce enough milk, probably, for the piglets to actually lactate or suckle. So then they were changed to a lactation diet. The piglets were weaned at day 28. We sampled blood and feces again of the sows. Then after the weaning, the sows were inseminated again for the second pregnancy. So that was only partly a pregnancy until day 70. So again, at day 30 of the second pregnancy, we also sampled blood and feces of the sows. And at day 70, we euthanized the sows and we sampled the fetuses. So then we measured the short-chain fatty acid levels of the feces. There's a lot of data in this figure. So here is the levels of acetate, which, as you can see, are much higher than the other 2. Propionate is a bit lower than acetate, and butyrate is rather low, which is actually known. Butyrate is always the lowest to measure. So the different time points that we had: time point 1 before gestation, time point 2 is day 30 of the first pregnancy, time point 3 day 70 of the first pregnancy, time point 4 during lactation of the sows, and then again in second pregnancy, day 30 and day 70. What you can see is an initial drop of short-chain fatty acids from the initial diet actually to time point day 30 of the first pregnancy. And this is because the diets that we actually gave them contains less fiber than the diet they are normally on. So that leads to a drop of short-chain fatty acid levels. And basically what you can see from this figure is that the levels usually don't vary that much during pregnancy or during the pregnancy. Then on time point 4, you see an increase again, and this is due to the lactation diet that the sows were on, which also contains more fiber to be able to produce, well, not only more fiber, but also it's a very rich in nutrient diet, because of course they need to produce enough milk. And And then you see a decrease again based on our intervention diet. Mainly in the high fiber P diet, so that's the red one, you see that there's quite some variation in acetate. We currently do not really have an explanation for that. We're still working out all the results, so it's not a clear-cut story yet. Yeah, so these were the fecal short-chain fatty acid levels. Then if we look at the serum acetate levels in the sows, we see that they're much lower. Also, they're less variable. There's a smaller difference between the different diets. So again, here the same time points. And here we only look at the acetate levels because in serum, it was very hard to measure propionate and butyrate because they were around a threshold level. Basically, you see the same thing here in time point 0 when they are on a different diet, the levels are much higher, and you see a decrease when they are transferred to our experimental diets or intervention diets. The difference between the 3 different diets in the serum is not extremely high. And we've been thinking about that a lot, why that would be. And actually, as I will come back to in the conclusions as well, we think that maybe it's a timing issue, so that the short chain fatty acids level, I mean, so the samples that we take are actually from the periphery. And it might be that most of the short chain fatty acids are taken up by the liver and metabolized by the liver. So we cannot measure proper differences in the periphery. Also, the timing might be wrong. So maybe you need to measure like within 30 minutes after they've had their feed, for example. And this could not be timed because, well, this was how the experiment was done at the company. So this is, we could not really time specifically like 30 minutes after they had a diet to sample the serum, which we think is the main reason why we cannot see bigger differences between the 3 different diets. Then we also look at the fecal short-chain fatty acid levels in the 10-week-old piglets. Unfortunately, we did not sample the serum of those animals, so the only material we have besides organs is the feces. And actually here we do see a difference. So this is a one-time point, only at 10-week-old piglets. We see a significant decrease in acetate in the high fiber beet diet, and also significant difference between both high fiber diets. So there's significantly lower acetate in those piglets. And also we see some differences here in the propionate. And we think this could be due to the microbiota composition, so that the animals, if we go back here, the animals on the high fiber beet have a different microbiota. For example, here you see higher production in acetate. So they might have a different microbiota that actually accounts for a lower amount of acetate here in those piglets. At least that's what we now think is the proper explanation. So then if we look into the microbiota, we see that the fecal or the microbiota cluster based on the diet. So what you see here is a PCA plot, principal component analysis, where we actually looked at the genus levels, so the 16S of the piglets and the sows. Purple triangles are the piglets. You see they all cluster here nicely. And then the reds are the different, or day 0 of the sows, where actually all the sows are still on the same diet. And you can clearly see that there is a clustering, so the microbiota actually shifts as soon as they are on our intervention diets. And then if we do an RDA analysis, so this is what I depict here is day 30, but this plot looks the same for all different time points. And after we start the intervention, we see highly significant differences. So you can clearly see the difference between the control diet and the 2 other diets. The name actually, I should have adjusted this. So this is the low fiber diet basically, and this is the high fiber P, and this is the high fiber beet. So I apologize for the wrong names. This was, I forgot to adjust this in the figure. But you clearly see significant results after intervention, after the start of the dietary intervention. So at day 0, the p-value is 0.88. And then if you look at the days that the sows were on the diet, you see p-value highly significant. So you can clearly separate the sows based on the diet, which was actually what we were expecting to see. And then if we look at the piglets, we see that the microbiota also cluster significantly in the piglets on the RDA. So the p-value is 0.01. Although the separation is a bit less, but we also need to keep in mind that the piglets after weaning were all on the same diet. So the sows were on our experimental diets, but the piglets after weaning, they all got the same diet. So it's really nice that we can still see a separation, significant separation based on the diet the mothers were on. So that means that there is transfer of microbiota from the mother to the offspring in these piglets. So basically the conclusions here is that timing is very essential in measuring short-chain fatty acids in serum. We see that the short-chain fatty acid levels are quite variable in the serum, but that could be due to the timing issue. And we see the diet has a clear effect on the microbial composition, which is known from literature. So that was also expected, but it's nice that you see something that you expect also actually happening. So the take-home messages of today: short-chain fatty acids are major players in the interplay between diet, microbiota, and health. Short-chain fatty acid concentrations can be modulated by inclusion of non-digestible fibers in the diet. And don't forget to feed your own intestinal symbiont. So if we then go back to the hypothesis that I showed you in the beginning. So number one, the high fiber diet will increase serum concentration of acetate associated with changes in the microbiota. Well, partly because change in microbiota, yes, we saw increased serum concentrations. We think it was there, but we cannot prove it based on the assays that we've done or the samples that we have. And then the other ones, the maternal diet will have an effect on the fetus in utero via short-chain fatty acids exchange. There is an effect of genetic imprinting by the maternal diet, and that will have effects in the offspring up to 8 weeks postpartum. Serum acetate will regulate PPAR alpha and lipid metabolism, and serum acetate will upregulate fatty acid oxidation pathways in the liver and muscle, and protein metabolism in muscle. Well, I haven't shown any data based on these last 4 hypotheses, but hopefully Smahane will reveal all this in the follow-up talk. So then many people are involved in this big project. So from INRAE, we have Elisabetta, Hervé, Sarah, Andrea, and Smahane who all work on this project and did a lot of work on RNA-seq and ATAC-seq, as we will hear later on. Then on our side at Wageningen University, Jos Boekhorst has been helping me a lot with microbiota analysis, and Jerry Wells also for his scientific input. And then at Schothorst Feed Research, Francesc Molist, who basically led the whole animal trial, which we did at Schothorst. So these people have been very helpful and very necessary in this project. So thank to all of them. And thank you all for your attention.
Thank you, Linda, for your presentation. And everybody in the audience can send their questions to the chat box. And now I give the floor to Smahane. So she will present the influence of maternal diet on pig genome, epigenome and transcriptome. So Smahane, the floor is yours.
Thank you. Do you see my screen?
Yes.
Yes. Thank you, Linda, for the introduction. I'm Smahane Chalabi. I'm postdoc in bioinformatics and biostatistics in GenSwitch Web Package 5. And here I will present you the effect of the maternal diet on pig epigenome and transcriptome. Epigenome and its associated transcriptome vary between the different tissues and different cell types. Epigenome vary also during the developmental stage from gametogenesis until the adult stage. Epigenome is modulated by environmental factors like diet, and more particularly maternal diet, as described, as presented by Linda. Epigenome has the maternal diet have an impact on the epigenome, and multiple studies on livestock animals enable to revealed the effect of the maternal diet on the epigenetics, and this epigenetics effect may also impact the adult phenotypes. And epigenetic modification have an effect on the dynamic on the gene expression, an on-off dynamic. Our objective in the work package 5 is to better understand the effects of the maternal diet by the identification of the accessible regulatory region and the identification of the target genes. And we want also to assess the persistence of the epigenetic marks post-weaning. As well described by Linda, here is our experimental design, which has been performed by for which the sampling has been performed by the team of the Wageningen. We have 3 groups of mother pig with 3 different diets: a high fiber diet, high fiber P, plus acetate diet, and a low fiber diet. And we have 2 developmental stage, sample from 2 developmental stage, sample from piglets and from fetuses. The piglets and fetuses are from the same mothers. And we have similar number of male and females. And we have a sample taken from liver, which is a key metabolism organ, where small chain fatty acids are first metabolized, and samples from skeletal muscle. As explained by Linda, skeletal muscle has been shown as epigenetically modulated in previous study. We have also samples in biorepository and also preserved sample for generating organoids for further validation. And for some technical reason, not initially planned, we have 2 mother breeds and also 2 father breeds to take in account. Here is a summary of our experimental design. We have nearly 20 mothers, which follow 3 different diets. We have 2 developmental stages and piglets, a similar number of male and females, sample from 2 tissues, and mother breeds and father breeds to take into account in our model. We have nearly 300 samples that are well distributed between tissue stage and diet condition. In the work package 5, we have ATAC-seq data, which enable to identify regulatory accessible region, and RNA-seq data to identify the gene expression level. And ATAC-seq and RNA-seq data will enable to connect regulatory region to their target genes. The RNA-seq data will enable to have a quantitative view of the transcript expression. Here is an overview of the RNA-seq assay. We have first the sample preparation and the library generation, and then the RNA-seq library generation. And all the steps from raw pre-processing to different analysis have been performed by the team of Wageningen, and they have used the TAGADA workflow, which has been designed by the team of INRAE in CERN. The TAGADA workflow enabled to reconstruct gene and transcript and enable to generate a quantification of gene expression level. As you know, the chromatin accessibility is dynamic. Chromatin can be firmly impacted around nucleosome, so the chromatin is closed or inactive, and chromatin can be also nucleosome free. The chromatin is open or active. It enable the access for all transcription machinery for gene expression and gene expression regulation. ATAC-seq stand for Transophthase Accessible Chromatin with High Throughput Sequencing. This technology is based on two main steps, segmentation and amplification. During the segmentation step, the transposase insert into the open chromatin region. It cuts the DNA and insert small insert primers of sequencing, and then this primer, this fragment with the primers of sequencing are amplified by PCR and enable the generation of the library. Library are then sequenced, and in our project we have used the NF-core ATAC-seq workflow to translate reads into peaks, and then we have performed the differential analysis. In our work, we have used the Nextflow, which is a bioinformatics workflow manager, which enables the analysis of a large amount of data according to the FAIR principle, FAIR for fungible, accessible, interoperable, and reusable. And Nextflow has the particularity to be supported by NF-core community, which is a community effort which enables to collect curated set of analysis pipeline and offer all this quality pipeline to the community. Here it's an overview of the NF-core ATAC-seq pipeline. NF-core ATAC-seq pipeline is based on two main steps, the raw data processing and the data analysis. All along the pipeline, you have the quality control, a quality control for the sequencing, for the library, and for the functional annotation of peaks. Once reads are generated, are sequenced, they are submitted for a quality control, and then you have the trimming. Once reads are trimmed, they are aligned on the reference genome, and then you have two ways for the peak calling. You can merge you can merge reads from multiple library of same samples, or merge replicates in the same group. In this merging, you merge reads for a same group combination. For example, in our work, we have a different tissue stage diet group. You can merge sample from all sample from liver, piglet, diet, HF, high fiber, beets pulp diet together. And after each merging library, you have a quality control and also a step of data analysis, which generates a peak calling, consensus a peak set, also row count table for each sample. You have the number of reads observed at each peak region. And then you have the NF-core ATAC-seq pipeline generates a report in an HTML format. Here it's some quality control for ATAC-seq analysis. Here we have the read distribution profile in TSS region. In the x-axis, we have the position along the gene, and the 0 for the transcription start seat. And here you have the transcription end site. What we observe is that we have an enrichment at the upstream of the TSS and at the downstream of the TES, the transcription end site, which correspond to the regulatory region at the upstream and at the downstream of the transcription end site. the quality control gives you also the FRIP score, which corresponds to the fraction of reads in cold picked region. The FRIP score is the number of mapped reads which are under a cold picked region divided by the total mapped read. And in our work, in our project, we have a value around 0.3, which corresponds to a high proportion of read usable for PICS analysis. Here we have an overview of the statistical analysis method. We have first the step of the normalization for chromatin accessibility sites or ataxic data analysis, and also for the gene expression analysis for RNA-seq data. We have a step of normalization, and then we have a step of visualization with PCA, and then we can perform the differential analysis between all pairwise condition. And here it's our model for a given tissue and stage. We have different factors that we take into account for our differential analysis. The different factors are the diet, the mother diet, the sex, the mother ID, because we have different mother used in the project. We have the mother breeds and the father breeds. Here we have the global sample structure. The PCA revealed that we have for ataxic sample, for ataxic data, we have a clear separation on the tissue for the first principal component, and the second principal component separate clearly the sample according to the developmental stage, fetuses and piglets. The different colors correspond to the different diets in each tissue stage combination. The differential analysis revealed that differential peaks majority are clearly observed in muscle piglets. We have more than 650 differential peaks that are differentially accessible. Here is the annotation of the differential peaks, and we observe that most diet-specific differentially accessible regions are enriched in intron and promoter TSS compared to all consensus peaks. Considering RNA-seq data, the global sample structure is similar to the one observed in ataxic data. We have a first separation on the tissue, and a second separation according to the developmental stage. What we observed in the RNA-seq data is that we have no differential expressed genes. Then we have performed a gene set enrichment analysis, which revealed that which revealed enrichment of function, underlying metabolism, and immunity in muscle of piglets. If we read back to ataxic data, for which we have many differential peaks, we have exploited this results, and we have performed a transcription factor footprint analysis. In this figure, we have the DNA. The cycle in gray corresponds to the nucleosome, the cycle in blue to the transposase. The transposase can access into the open chromatin region and cut the DNA. And we have also some in some region, we can have some transcription factor. Here it has a hexagon in blue. And when we observe some sudden drop within the peak region of high coverage that correspond to footprints. These drops in the peak region correspond to the binding site of transcription factor. There are many tools that that enable to identify a transcription factor binding site, and in our work we have used hint attack, which is from the regulatory genomics toolbox, and this tool use open chromatin data to identify active transcription factor binding sites. Hint attack performed a first step of footprints calling and taking to input the bind file for which describe all reads for the different condition, and you give also the peak region, and it generates at first the predicted footprints into a bed file, and then you have the motif matching for the predicted footprints using the all motif from the database Jasper, Okomoko, or any other database. And then hint attack generates you motif predicting binding site and other profiles that can describe the binding site of particular TFF, transcription factor. So in our study, we were also interested in the differential TF activity to identify a tissue stage diet specific transcription factor. So as input, we have give motif predicting binding site that we have obtained in the previous step. For each, we give motif predicting binding site for each condition, and also reads from the bind file for each condition. And then hint attack gives you some tissue stage diet specific binding activity score for each transcription factor. Here we have a heat map of TF activity in fetuses and piglets for each tissue stage diets comparison. The described TF here correspond to differential active transcription factor in at least one of this combination. And I have marked with 1 star those with FDR lower than 10%, and with 2 star those TF with a differential activity lower than 5%. This heat map reveal some cluster of TF that need some more investigation. This TF, this diet-specific differential TF activity need some more investigation because they can reveal an underlying regulatory network of gene which may be orchestrated by those interesting TF. So this work is in progress, and we are working on it. As a conclusion on this work package 5, we have first performed a work on the data submission to Funk Data Coordination Center. We have submitted more than 600 FASTQ files for the data portal. concerning the ataxic data, we have a few differential peaks, but they are enough significant. And they are triggered by high fiber maternal diet composition, mostly in piglets' muscle. For renisic data, the GSEA analysis show function underlying metabolism and immunity. We have some perspective to this work. We're interested in linking the differential TF activity footprints to gene expression, to decipher the underlying regulatory network. And we are also interested in the integration analysis of all the previous data presented by Linda, such as serum microbiota and this renisic and ataxic data to gain biological insights. And this work is also in progress. To finish, I want to acknowledge all GenSwitch partners, and more particularly the Work Package 5 partners. And thank you for your attention.
Thank you, Smahane, for your presentation. So if there are any questions, please feel free to write them on the chat box. But while waiting for your questions, we have some questions for Linda. So if Adevunmi is still online, maybe you can directly open your microphone and interact with the presenters and ask your questions.
Yeah, I saw that there was one question in the chat box indeed. Whether there are predefined biomarkers for short-chain fatty acids in jejunum of chickens. To be very honest, I don't know much about, or basically anything about short-chain fatty acids in chickens. So I'm very sorry, but I cannot say anything about chickens. The other part of your question, I can say something about it, whether short-chain fatty acids are restricted to the microbiota response in the colon only. Well, the main fermentation indeed happens in the colon. So in the small intestine, there is some uptake by the intestine itself. And the fermentation really takes place in the colon because that's where the main or the highest load of bacteria are. Yeah. So whether you could identify short-chain fatty acid signals in the duodenum jejunum, I'm not aware of any literature that actually looks at, well.
Yeah, the question basically.
Oh, sorry.
Yeah, the question basically is focused on the fact that, yes, we can actually identify microbiota also in the jejunum, yep, or the ileum or the duodenum. Now, is it possible in any way we can, of course, have biomarkers for the function, the potential functions of this microbiota to produce SCFA, even if, of course, yes, I agree with you, the load of the fermentation, of course, occurs at the distal part of the GIT. In the chicken, for example, more experiments are in the cecum. But my interest, of course, is in the absorption of nutrients. We want to, in a way, link the effects of microbiota to the interplay of absorption of nutrients. And then, yes, while we cannot necessarily pin the function of the microbiota to utilization of minerals in the GIT, could it be that the SCFA might be one of those proxies or the functions of the microbiota? And this is why I'm focused more on this part of the absorption of nutrients and not where we have the load for microbiota. And that was why I asked the question.
Yeah. Well, there are some levels also. Also some short-chain fatty acids are produced by the host itself, but that's very limited if you compare to what's produced by the microbiota. So there are some levels indeed in the small intestine, but that's much lower because of the lower microbiota load than in the colon. So I'm not sure if you can well, as you say in the question, to identify the short-chain fatty acid synthesis signals. You can, of course, like take a, when you, for example, dissect an animal, you can at that point take samples from the small intestine, measure short-chain fatty acids in the luminal content of that part that you're particularly interested in, and also look at the microbiota composition. And in that way, you could try to link indeed the short-chain fatty acids that are present to the microbiota that is present at that specific location. But then that would need, like, that's an end point measurement, because I mean you need to actually access those samples at that moment. But then I think if you compare the short-chain fatty acid being present in the lumen to the 16S of the microbiota, you might be able indeed to link those two together. However, the levels will be much lower indeed than in the colon. Does that answer your question?
Oh, yeah, thank you.
You're welcome.
Thank you, Adewunmi, for your question.
You're welcome.
Are there any other questions for Linda or Smahane? Or maybe about the program, European Master of Animal Breeding in Genetics to Gabor? Okay, maybe I can ask a question. Well, I don't know how many of the participants are on. Oh, I have something here. So Yusuf, maybe you can just ask your question, open your microphone, and if you'd like. Oh, okay. He doesn't have a microphone. Well, after obtaining the differentially accessible peaks, have you annotated the peaks and performed functional enrichment analysis? What tools have you used for it? And yeah, there's a second part of the question, Linda. Maybe you see it already.
Yes, the question I think is for me. So the question is, have you annotated the peaks and performed functional enrichment analysis? Yes, in fact, we have performed this work for the tool used for the annotation. The annotation of the peak, we have used Homer. And for the functional enrichment analysis, we have used, we first performed GSEA also for ataxic data, but we prefer to continue to follow the analysis on a hint attack or TF footprinting analysis, and we've used this functional enrichment analysis more on RNA-seq data. So we've used here GSEA. The second question is, you have found a small number of differentially expressed peak in attack seq analysis. Why do you think that you haven't found a significant result for the RNA-seq? We have no differential gene expressed in RNA-seq data, but to connect our ATAC-seq data to RNA-seq data, we will use this hint attack results, which give us footprinting, predicted footprinting to connect the interesting TF that are differentially expressed for each usage stage diets comparison, to connect this TF to RNA-seq gene expression data. The third question, do you have a plan to do single cell ATAC-seq as well? Yes, it's in discussion, but for another project. I can tell you more about that. It's not in the Work Package 5 Gen Switch project. Is it clear, Yusuf, for the response to your question?
Well, he doesn't have a microphone, so maybe.
Hello, can you hear me?
Yeah. Yes.
I am in the same office with Adewunmi.
Okay.
So here is a chance to see me. I am Yusuf. Yes. I am doing a PhD and I'm doing also the same thing, a toxic analysis in phosphorus in chickens and hens. And yeah, I was expecting for you to answer about RNA-seq analysis that I didn't get clear idea of the answer because you have differentially expressed peaks, so that I expect to then the genes must be expressed for me in my ideas, and then you looked into RNA-seq, and then now there is nothing. So maybe do you have
genes are expressed, but are not differentially expressed for each tissue stage diet combination. And I don't know if I answer well to the question or not.
Yeah, most of it.
Okay. You have another question?
I will follow up later.
Okay, thank you. Do not hesitate to contact me or to ask me other question if you need.
I am really interested. Okay.
Thank you, Yusuf. So if there aren't any other questions, maybe we can close the meeting. I just have one last slide to show you. And yeah, so if you are interested more in our project and EMABG, you can always contact us through the social media channels or visit our website to get more information. And also you can just subscribe to our newsletter for GeneSwitch, or you can also contact our coordinators. Thank you very much for joining today. And we would also like to thank Gabor and the EMABG team, Natalie, for giving us this opportunity. Have a nice day. I don't know, Gabor, would you like to say anything as a closing?
No, I think it was well rounded up, and so there are kind of contact points and contact possibilities. So if you are interested, we are just more than happy to follow up any inquiries that you might have.